Welcome to A to Z Nursing Vlogs. This video is about a group of related RNA viruses that cause diseases in mammals and birds called as the coronavirus. Let's dive into the video. To introduce this topic, coronaviruses are a group of related RNA viruses that cause diseases in mammals and birds. In humans and birds, they cause respiratory tract infections that can range from mild to lethal. Mild illnesses in humans include some cases of common cold, which is also caused by other viruses, predominantly rhinoviruses. More lethal varieties can cause SARS, MERS, and COVID-19, which is causing an ongoing pandemic. In cows and pigs, they cause diarrhea, while in the mice, they cause hepatitis and encephalomyelitis. The constitution of this coronavirus is that it belongs to the subfamily Orthocoronavirinae, in the family Coronaviridae, order Nidoviralis, and realm Riboviria. They are enveloped viruses with a positive sense single-stranded RNA genome and a nucleocapsid of helical symmetry. The genome size of the coronaviruses ranges from approximately 26 to 32 kilobases, one of the largest among the RNA viruses. They have characteristic club-shaped spikes that project from their surface, which in electron micrographs create an image reminiscent of the solar corona from which their name derives. The interesting etymology is that the name coronavirus is derived from Latin word corona, meaning crown or wreath, which itself is a borrowing word from the Greek, which is from corone, meaning garland or wreath. The name was coined by June Almeida and David Tyrrell, who first observed and studied human coronaviruses. The word was first used in the print in the year 1968 by an informal group of virologists in the journal Nature to designate the new family of viruses. As the number of new species increased, the genus was split into four genera, namely alpha coronavirus, beta coronavirus, delta coronavirus, and gamma coronavirus in the year 2009. The common name coronavirus is used to refer to any member of the subfamily Orthocoronavirinae. As of the year 2020, 45 species are officially recognized. The structure of coronavirus. Coronaviruses are large, roughly spherical particles with unique surface projections. Their size is highly variable with average diameters of 80 to 120 nanometers. Extreme sizes are known from 50 to 200 nanometers in diameters. The total molecular mass is on average 40,000 kDa. They are enclosed in an envelope embedded with a number of protein molecules. The lipid bilayer envelope, membrane proteins, and the nucleocapsid protect the virus when it is outside the host cell. The viral envelope is made up of a lipid bilayer in which the membrane envelope and the spike structural proteins are anchored. The molar ratio of ESM in the lipid bilayer is approximately 1 is to 20 is to 300. The E and M protein are the structural proteins that combined with the lipid bilayer to shape the viral envelope and maintain its size. The S proteins are needed for interaction with the host cells. The diameter of the complete envelope is about 85 nanometers. The life cycle of coronavirus takes place by 6 important steps. Number 1. With the S protein, coronaviruses bind on cell surface molecules such as the metalloprotease, aminopeptidase N. Viruses which accessorily have the HE protein can also bind on N-acetyl neuraminic acid that serves as a co-receptor. Number 2. So far it is not clear whether the virus gets into the host cell by fusion of viral and cell membrane or by receptor-mediated endocytosis, in that the virus is incorporated via an endosome, which is subsequently acidified by proton pumps. In that case, the virus have to escape destruction and transport to the lysosome. Number 3. Since the coronaviruses have a single positive stranded RNA genome, they can directly produce their proteins and new genomes in the cytoplasm. At first, the virus synthesizes its RNA polymerase that only recognizes and produces viral RNAs. This enzyme synthesizes a minus strand using the positive strand as a template. Number 4. Subsequently, this negative strand serves as a template to transcribe smaller subgenomic positive RNAs, which are used to synthesize all other proteins. Furthermore, this negative strand serves for replication of new positive stranded RNA genomes. Number 5. The protein N binds genomic RNA and the protein M is integrated into the membrane of the endoplasmic reticulum, like the envelope protein S and He. After binding, assemble nucleocapsides with helical twisted RNA butt into the ER lumen and are encased with its membrane. At last, number 6, these viral progeny are finally transported by Golgi vesicles to the cell membrane and are exocytosed into the extracellular space. This is the life cycle of the coronavirus. The transmission. Infected carriers are able to shed viruses into the environment. The interaction of coronavirus spike protein with its complementary cell receptor is central in determining the tissue tropism, infectivity, and species range of the released virus. Coronaviruses mainly target the epithelial cells. They are transmitted from one host to another host depending on the coronavirus species by either an aerosol, fomite, or fecal oral route. Human viruses infect the epithelial cells of the respiratory tract, while animal coronaviruses generally infect the epithelial cells of the digestive tract. SARS coronavirus, for example, infects the human epithelial cells of the lungs via an aerosol route by binding to the angiotensin converting enzyme 2 ACE receptors. The transmissible gastroenteritis coronavirus infects the pig epithelial cells of the digestive tract via a fecal oral route by binding to the alanine aminopeptidase receptor. Here goes the classification. Coronaviruses form the subfamily Orthocoronavirinae, which is one of the two subfamilies in the family Coronaviridae, order Nidoviralis, and the realm Riboviridae. They are divided into four genera: alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. The alpha coronaviruses and the beta coronaviruses infect the mammals, while the gamma coronaviruses and the delta coronaviruses primarily infect the birds. Here we see the phylogenetic tree of the coronaviruses. Here we see the origins of human coronaviruses with possible intermediate hosts. Bats and birds as warm-blooded flying vertebrates are an ideal natural reservoir for the coronavirus gene pool, with bats the reservoir for alpha coronaviruses and beta coronaviruses, and the birds the reservoir for the gamma coronaviruses and the delta coronaviruses. Many human coronaviruses have their origin in bats. Unlike other beta coronaviruses, bovine coronaviruses of the species beta coronavirus 1 and subgenus Embicovirus is thought to have originated in rodents and not in bats. The MERS-CoV emerged in humans from the bats through intermediate host of camels. It is speculated that the flu pandemic of 1890 may have been caused by this spillover event and not by the influenza virus, because of the related timing, neurological symptoms, and unknown causative agent of the pandemic. Besides causing respiratory infections, the human coronavirus OC43 is also suspected of playing a role in neurological diseases. In the 1950s, the human coronavirus OC43 began to diverge into its present genotypes. Infection in humans. Coronaviruses vary significantly in risk factor. Some can kill more than 30% of those infected, such as MERS-CoV, and some are relatively harmless, such as the common cold. Coronaviruses can cause colds with major symptoms such as fever and a sore throat from swollen adenoids. Coronaviruses can cause pneumonia, either direct viral pneumonia or secondary bacterial pneumonia, and bronchitis. The human coronavirus discovered in the year 2003, SARS-CoV, which causes severe acute respiratory syndrome, has a unique pathogenesis because it causes both the upper and the lower respiratory tract infections. 6 species of human coronaviruses are known, with one species subdivided into 2 different strains, making 7 strains of human coronaviruses altogether. Of the 7, 4 human coronaviruses produce symptoms that are generally mild, even though it is contended they might have been more aggressive in the past. Number 1, human coronavirus OC43. Number 2, human coronavirus HKU1. Number 3, human coronavirus 229E. Number 4, human coronavirus NL63. Three human coronaviruses produce potentially severe symptoms. Number 1, severe acute respiratory syndrome coronavirus SARS-CoV, which is identified in the year 2003. Number 2, Middle East respiratory syndrome related coronavirus identified in the year 2012. Number 3, severe acute respiratory syndrome coronavirus number 2, which is identified in the year 2019. These causes the diseases commonly called the SARS, MERS and COVID-19 respectively. The prevention and treatment methods of coronavirus. A number of vaccines using different methods have been developed against human coronavirus SARS-CoV-2. Antiviral targets against human coronaviruses have also been identified, such as viral proteases, polymerases, and entry proteins. Drugs are in development which target these proteins and the different steps of viral replication. Vaccines are available for animal coronaviruses IBV, TGEV, and canine CoV, although their effectiveness is limited. In the case of outbreaks of highly contagious animal coronaviruses such as PEDV, measures such as destruction of the entire herds of pigs may be used to prevent transmission to other herds. If you like the content of this video, like the video and share this with your friends and colleagues. Subscribe the channel and click on the bell icon for more videos and updates. Thank you.